Well, welcome everybody. I have about 2 minutes after, so we're going to go ahead and get started. This is the third in the series of 3 horse management seminar webinars for the spring, I almost said fall, spring 2022 season. Thank you very much for joining us. I am super excited about today's speakers, and we'll talk more about them in just a minute. So I'm Dr. Carrie Williams. I'm the Equine Extension Specialist here at Rutgers. Been here for going on 19 years now. So this is definitely not my first horse management seminar, but I will say it's only the second virtual horse management seminar that we've had. We had ours last year due to COVID, and this year we just decided things weren't quite stable enough, especially at Rutgers, to go live. But hopefully next year we'll be back in person, but I think we've had such great responses to these horse management seminars virtually that we might possibly continue them as well. So, and with that being said, I just saw us tick over 100 attendees. So thank you again for joining us. Before we get too much into the speakers, I have a short little video clip. If you haven't been in attendance at any of our Equine Science Center events, I would like to share that with you, because this event is not just a part of the Rutgers Cooperative Extension System, but by my appointment, I am the Assistant Director of Extension, for the Equine Science Center. So let me just share a little bit about the Equine Science Center with you.
Good morning. Welcome to the George H. Cook campus, home of the New Jersey Agricultural Experiment Station, the School of Environmental Biological Sciences, and of course, home to the Rutgers Equine Science Center. The Equine Science Center is a soft-walled center where we bring together the best and the brightest to solve challenges impacting horses, horse well-being, and the horse industry. To provide you more details about the research being conducted at the Equine Science Center, and specifically the Equine Exercise Physiology Lab, I'd like to introduce Dr. Kenneth McKeever.
The lab was founded in 1995, and since then it's been one of the most productive research labs in the country, if not the world. We've conducted studies looking at how to care for the aged horse. That's a horse that's over 20. We've looked at various drugs that affect performance. We've even done studies for the Army looking at food extracts that can reduce inflammation. So the Equine Science Center and the Equine Exercise Physiology Lab are a valuable asset to how we care for horses in the state of New Jersey.
So we've all visited this morning the Equine Exercise Physiology Lab, part of the Equine Science Center at Rutgers. We have another facility where cutting-edge research is being carried out, located on Riders Lane, and it is the site of the first of its kind environmental best management practices demonstration horse farm, located here on the George H. Cook campus. The team of scientists who are helping horse farmers become better environmental stewards and caretakers of the land is a team of multidisciplinary faculty, students, and staff led by Dr. Carrie Williams, Dr. Mike Westendorf, PhD student Jennifer Weinert, and other faculty, students, and staff from different departments here at Rutgers and beyond.
Hello, I'm Dr. Carrie Williams, the Equine Extension Specialist here at Rutgers University and the Associate Director of Extension for the Equine Science Center. We are here at the Environmental Best Management Practice Demonstration Horse Farm. This serves as a wonderful venue for the public, farm owners, other industry professionals to come look and see the 8 various best management practices, educational venues that they can use on their farm or in their areas. It is open to the public. You can come anytime. This venue also serves as a great location for research. My current graduate student, Jennifer Weinert, is looking at both cool season and some novel warm season grasses to see the production as well as the horse's metabolism. Some glucose and insulin responses as well as the microbiome will be looked at over the next 2 years of this project. Thank you for visiting all of us at the Equine Science Center. Our website can be found at esc.rutgers.edu.
case you aren't familiar with the Equine Science Center. Some of those names we're going to revisit in just a little bit. But I want to mention that even though this program is free for all of you to attend, there are a lot of things that we do that aren't free, namely all of our research. We've got a huge amount of research that goes on at the Equine Science Center, and a lot of it can't be funded without people who help us financially in terms of donations. So we just ask if you are at all interested in helping with the research that we do. Any amount would be wonderful, even if you have $2 that you want to say you gave for part of some of this research. We'd love to have you donate. The link is at the bottom of your screen right now. Our Equine Science Center website has a donate page. We'd love to have you think about donating. We also have a really nice memorial page. You can donate in someone's name or even your horse's name. I know I've done that in the past. So that was a little bit about us. Now, what I want to do is know a little bit about you. So what you see on the screen now is something called Menti. And if you've been with us in the last 2 seminars, you'll know exactly where to find this. But everybody, if you go to your phone or another web browser or some way that you can type in menti.com, that's all you need, MENTI.com. And I'm going to type it in the chat for everybody. Once you get into Menti, you'll enter a code, and that code is also at the top of this. And I see some people are already jumping on in there. I'm going to make this full screen so we can see it better. But this code that you're going to need is 88231133. So that's also in the chat. So I'm going to give you a little bit of time for those who are just logging on. I just did a welcome about us, and now I want to welcome about you. So it's always fun to see this little word cloud change. If you enter in your state or your country, please type in the whole name, don't abbreviate. And the reason for that is because the more people that actually say that word, the bigger the word is going to get. So right now you see New Jersey is very large. That's about right, because a lot of the clients that I use in all of our blast lists are from New Jersey. But we also have a good contingent in Pennsylvania. Michigan and North Carolina aren't far behind. I see Virginia creeping up as well. So I'm going to leave this change a little bit because I see a lot of people are still logging on. And it's great to have you all. Looks like we're at about 145 of you attendees now. So thank you so much. And if you are just logging on, we're going to something called menti.com. So if you go to menti.com and you type in that code at the top, it's 88231133. I just want to know a little bit about where the demographic is. And then when I change the slides, we're going to learn a little bit more about what you do and how you're involved with the horse industry. The other fun thing is last year, we went through and we analyzed where everybody was coming from in terms of states and countries. And we actually had 39 states represented last year at our horse management seminar. And I'm really interested to see what states are going to be represented this year, or how many states this year. And also want to see how many countries and what countries. So if I can follow all the changing words, right now I see Brazil and Peru and Mexico. So welcome all of you internationals. I also see, oh, there, Ireland. Welcome, Ireland. Alaska isn't another state, but you're pretty far, or it isn't another country, but you're pretty far away. So welcome from Alaska as well. So we still have New Jersey and Pennsylvania on top, followed closely by North Carolina, Georgia, Virginia, Michigan. And I see it's still even changing with 82 people logged on. Oh, Canada, I see popped on there. It takes a while to like get your eyes to focus on where all the words are going. Awesome. Well, I'm going to leave that. Keep going. If you could log on late, you can still log on. But let's go to the next one, because the next slide is just asking what your primary interaction with horses is. And I actually let you select more than one, so that if you do own horses, but you also train, you can pick those too. But, you know, just kind of your primary interactions. Because I know pretty much if you are a horse owner, you're also an enthusiast, but the enthusiasts are there for those of you who might not own horses, but are just interested in the material that we have for you. So again, if you're just logging on, we're going to menti, MENTI.com, and typing in 88231133, and that's going to give us a little bit of information about who our audience is and who we're talking to today. I feel like this is also good for the speakers, just to get a little idea. If you are one of the 14 others, you can feel free to type in the chat box and tell us what other you might be. I know in the past we've had farriers, therapeutic riding instructors, massage therapists, you name it. Oh, Equine Science Center students. So welcome from another university, I'm sure. A farrier, some path instructors, great. Had a performance at Triple Crown Nutrition, welcome, thank you. And a nutritionist, yay, nutritionists out there. This is the day for you, we got a lot of nutrition. But as always, we usually have quite a few horse owners out there. It looks like we got quite a few of you. Also some bunch of enthusiasts, trainers, barn managers, other industry professionals, whether you're pharma, feed, tack, etc. A couple of veterinarians logging on. And a bunch of others. So more others, some volunteers at therapeutic riding centers. I think I actually need to put therapeutic riding as a category next time, because I've always had so many of you in the therapeutic riding area. That's great. Horse show announcer, welcome. Awesome. So this is really, I love watching it change and go, but I won't take up too much of your time with this, even though I do really get a kick out of where everybody's from and what you're doing. So so thank you so much. If you are logging on late, I'll definitely take your answers. This will keep on changing until I completely close it out, which probably won't even be until tomorrow. So I'm just gonna go ahead and stop sharing. I've got a little bit of tidy housekeeping, I think we usually call it, stuff before I go ahead and introduce our first speaker. So first off, we are going to be primarily using our Q&A box for our questions. So while I did have you type in chat, if you do have any technical difficulties, or you just want to talk to the panelists, you can go ahead and enter it in the chat. But what I would really hope is if you have any questions for our speakers today, right next to the chat box, if you kind of wiggle your mouse over the bottom of your screen, you can see there's a little Q&A bubble. We're going to use the Q&A bubble for answering questions. First, it helps me moderate them. I see them easier, and I don't get lost in the huge chain of chats. And if they are answered online, everybody will also be able to see the answers. So please go ahead and use that. What we're going to do is we're going to listen to the first speaker. And then I might take one or two questions for in between, depending on time. But what I really want to do is wait until the very end after both of our speakers, and then open it up for Q&A for both of them. Because while you might have specific questions about exactly what they were talking about, you might also have some other questions that more of us might be able to answer. So with that being said, I've covered all of my intro material other than to introduce our first speaker. And when I start doing that, I'll ask Dr. Weinert-Nelson to come on the screen, and she can go ahead and start sharing. So you might have already heard her name, but you've heard me call her Dr. Jennifer Weinert or Jennifer Weinert, but she is now Dr. Jennifer Weinert-Nelson. So as you heard in the video, and I can jump to the end of her little bio she gave me, which I probably don't even need to read off the screen. Dr. Weinert-Nelson was my graduate student for the past 5 years. She came to Rutgers in the fall of 2016, and she just graduated with her PhD in September. Well, officially in January, but she defended in September of 2021. I was very sad to see her go, but she just started a very fun and educational position, which I think she's going to learn an awful lot at the USDA, their Agricultural Research Service Center, and their forage and animal production unit in Lexington, Kentucky. She's been there about a month now, maybe just about a month. But prior to that, so now I'll go back to the beginning of her intro. She grew up on a dairy farm in northern Wisconsin. She got her bachelor's degree in animal science from the University of Wisconsin River Falls. She then went to work in the Western performance sector, doing a lot of reining horse training, both in Texas and Wisconsin. And she got a real great appreciation for the equine industry. But then she ended her time in industry and reached back out to UW River Falls. and ended up at their campus laboratory farm, where she oversaw a lot of the university or all the university teaching horses. She helped with classes, she did outreach programs, she also managed their breeding farm. So then I stole her from the Rubber Falls to further her education. She is a great teacher, a great speaker, and really the theme of today, which I am really excited about, is recent advances. That was kind of the theme for everything. And what I wanted to do is really get to the bottom of a lot of the recent advances that we've uncovered with equine nutrition. And I will say Dr. Weinert-Nelson's PhD was absolutely massive. So I only asked her to focus on a tiny piece. And that deals with a lot of the grazing work that she did, as you heard in the video, metabolism of the grazing horse, and the microbiome, because there is a very hot buzzword nowadays. So she's going to explain a little bit more about that and uncover some of her research. So I'm going to stop talking and turn it over to you.
Well, thank you for that introduction, and thank you, Dr. Williams, for having me back. I'm happy to be a part of the Rutgers Horse Management Seminar Series, this time around as a speaker. And for all of you at home who've taken the time out of your evening to log on, thank you for joining us. As Dr. Williams said, tonight my talk is going to focus on that relationship between metabolism and the hindgut microbiome in the grazing horse. But before we get to that, I think it's really important that we all understand some of those microbiome basics, some of the basic things about what even a microbiome is, so that we can start to understand some of the research and how we might apply that. So we're going to start by asking you a question. And the question is, where is the microbiome? Is it in the gut, on the skin, in the ocean, on the surface of your cell phone, or all of the above? Now, we don't have a polling feature here, so all you just think about this on your own at home and kind of give this a couple of seconds of thought. And while you're thinking about this, what I'm going to tell you is that before we really can answer this question of where is the microbiome, we maybe need to know, well, what even is the microbiome? And to answer this question, we have to first acknowledge that this probably isn't even actually the correct way to ask this question. So instead of what is the microbiome, what we really need to ask is what is a microbiome? Because the definition of a microbiome is simply a community of organisms, or more technically their genes, that inhabit a particular environment. I'll say that again. A community of microorganisms that inhabit a particular environment. Any particular environment. So yes, it is true that there is a microbiome in the gastrointestinal tracts, or in the guts, of humans and animal species, including the horse. But even within the gastrointestinal tract, different parts of the GI tract will have different microbiomes. So for instance, the microbiome in the mouth is different from the microbiome in the small intestine, is different from the microbiome in the large intestine. And if we look at the horse, within the horse's large intestine, there is a different microbiome in all of the different compartments of that large intestine. For example, the microbiome in the cecum differs from the microbiome in, say, the large colon. And in all animals, the microbiome exists not just in the GI tract, but in other parts of the body and on the body, yes, on the skin. We also can find microbiomes in environmental samples. So there's a soil microbiome, there are microbiomes in bodies of water, and inanimate objects even can have microbiomes. So there's a microbiome that lives on your doorknob, or maybe on your mouse or keyboard of your computer. So when we go back to this question, obviously the answer is all of the above. There could be a microbiome in any one of those places. But for purposes of our presentation tonight, we are specifically interested in the equine hindgut microbiome. So what is the hindgut? To understand a little bit about gastrointestinal anatomy in the horse, I like to think of the horse's GI tract in two big pieces. So we've got the foregut, which is basically everything from where feed comes into the mouth, through the esophagus, the stomach, and the small intestine. And this foregut is where nutrients like sugars and starches and proteins and fats are primarily broken down and absorbed by the horse. However, the enzymes that horses and actually all mammals produce are not capable of breaking down the fiber component in the diet. And therefore, the fiber passes into the hindgut, or large intestine, where it's subject to fermentation by the large and active microbial population that lives there. So what is this fermentation process? So I said, fiber passes into that hindgut and it is subject to microbial fermentation. So the microbes can break down that fiber and they use that fiber for growth and reproduction so they can make more cells and get bigger. However, I think it's important to point out, you see I have over here fiber and. So we always think of this fiber, and fiber is the biggest part of fermentation in the hindgut. However, it should be noted that bacteria can live on just about anything. So in addition to fiber, there are also bacteria that are capable of breaking down other nutrients, like sugar, starches, protein. And there are a certain amount of sugar, starches, protein in the horse's diet that for whatever reason escape digestion in the foregut and then would be in the hindgut along with that fiber and would be digested by the microbes that live there. And what happens during that fermentation process? Yes, the microbes use those nutrients to grow and reproduce, and they also produce a lot of byproducts as a part of that fermentation process. And these are things like short chain fatty acids, or SCFAs, B vitamins, vitamin K, and some gases. And of these, the most important are probably these short chain fatty acids. Now, just as a note, you might have heard of volatile fatty acids or VFAs, SCFAs, VFAs, those terms can kind of be used interchangeably. For terms, or for this presentation, we're going to use the SCFA, but just know that if you see that somewhere, we're pretty much talking about the same thing. And why is this important? Why are these short chain fatty acids so important? Well, once these are produced as a byproduct of fermentation in the hindgut, they're absorbed by the horse and they're used by the horse for energy. In fact, short chain fatty acids that are produced from bacterial fermentation in the hindgut actually account for something like 50 to 70% of the horse's daily energy requirement. 50 to 70%. So the horse is getting a lot of its energy from that fermentation process in the hindgut. So let's narrow down a little bit more on just that grazing horse microbiome, the hindgut microbiome of the grazing horse. So why do we care? If you are a producer or owner sitting at home, why should you learn more about this? Why would you be interested? What could you benefit from having a better understanding of the hindgut microbiome of your horse that you maintain on pasture? Well, we just said that fiber is the big part of fermentation in that hindgut. And forages like hay and pasture are high fiber feeds. There's a lot of fiber in our pastures, and therefore the hindgut microbiome is going to play a big role in how that fiber is utilized by the horse. So what are some questions that we might want to answer, or some areas that we might want to know more about? Well, obviously, we're probably going to want to know a little bit more about how those hindgut microbes utilize nutrients from that pasture forage. We might also be interested in learning about any potential microbiome dependent impacts on the metabolism of the grazing horse. And we're probably going to be interested in how changes in pasture forage could potentially affect the hindgut microbiome. We know that there are changes, say, in nutrient content of pasture forages as we move across the growing season. There are also changes in forage availability, or we might be just interested in how horses' microbiomes are impacted by different types of pasture forages. And then moving on from that, we really want to know, is there any influence on horse health outcomes? Does that microbiome influence things like the weight gain that we see on pasture, or the development of things like metabolic syndrome or insulin resistance and laminitis? And finally, what the really important part is that we want to be able to use what we learn about these areas to inform our management choices and potentially identify interventions that we can use to either treat or hopefully prevent maybe some of these more negative health outcomes. If we look at the research or the body of work that's been done around grazing horse microbiome, there's really covered three primary areas of study. So the first of which is inter-diet comparisons. So comparisons in the hindgut microbiome of horses maintained on pasture versus the hindguts of horses that are fed a hay diet or a mixed diet that contains concentrate component, or maybe differences between different pasture forages. And beyond that, some studies have started to look at those seasonal changes that occur across a grazing season. And more recently, there's been a beginning to look at this topic of relationships between the microbiome and the metabolism of the grazing horse. However, of all of these, by far, the most work has been done on number one here, on these inter-diet comparisons. So this is kind of where the bulk of the work has been conducted thus far. And when we talk about inter-diet comparisons, research that look at these kind of approach this in two different ways. So perhaps they are interested in differences that occur after adaptation to different diets or different forages. So horses that have been maintained on pasture for an extended period of time versus the hindgut microbiome of horses that have been maintained on a mixed diet that has concentrate in it. But in addition to wanting to know what happens after horses are adapted, it's probably also interesting and important to know what happens as horses transition between diets. What are some potential fluctuations that we might see in the hindgut microbiome of horses as they transition, say, from pasture forage to hay? In these studies of the equine hindgut microbiome, pretty much all of them will start with evaluating what's called diversity. And they'll evaluate potentially a couple of different kinds of diversity, but they always pretty much start with this concept of alpha diversity. And here we're really talking about the species richness, or maybe the species richness and evenness of the horse's microbiome. And in this study, they evaluated the hindgut microbiome of horses grazing pasture grass, and then horses fed silage and a hay diet. So obviously this wasn't conducted in the United States. Here we don't really use silage as a form of forage for our horses, but in other parts of the world, it is a commonly used forage source. And what the study found was that the alpha diversity, the species richness and evenness of horses that were maintained on this pasture diet, represented by P in this figure, was greater than horses that were fed either the hay diet or the silage diet. And so pasture potentially results in a more diverse hindgut microbiome. The second type of diversity that's usually evaluated in these studies is something called beta diversity. And this is oftentimes presented in these PCOA plots. That's what these kinds of plots are called. And really, what you need to know when you look at these is, how do I interpret this? Because they can be really confusing if you don't know how to read one of these plots. And so anytime you see one of these plots, every point is going to represent a sample, or in this case, a horse. And these are often color-coded by diet. In this case, horses that were maintained on pasture are shown in the blue triangles, horses that were fed the hay diet in the red circles, and horses that were fed the silage diet in these green diamonds. And what we're looking at in these PCOA plots, what we want to see if there is a difference in diet, or if this is a different study, a difference in whatever variable you're studying, is that you want to see clustering of your points by diet or by whatever variable you're assessing. And so in this case, we are seeing that. So here it's a pretty clear clustering of these horses that were maintained on pasture, these blue triangles in our lower right-hand corner of our plot, versus our hay diet horses that are over here in the lower left-hand corner in our silage diet that's up here. And this really isn't telling us that it's more or less diverse, but what it is telling us is that there is a distinct microbial structure And then the structure and composition of that hindgut is different in horses that are maintained on pasture versus horses that are maintained on a hay diet or silage. But it's not just enough to look at diversity. Then we have to move beyond that and look at the species composition of the hindgut. So we may look at different levels of taxonomy, or we might look at specific bacteria that we're really interested in. In this study that I'm showing you here, they looked at horses that were maintained on a grass pasture versus horses that were fed that mixed diet with a concentrate component. You don't need to worry about this SCOD. That was another condition that they were studying in this particular research study. But here, just focus on these grass and concentrate horses. What they did find was that species in the genus Fibrobacter and species in the family Ruminococcaceae did decrease when horses were maintained on a concentrate diet versus when they were maintained on pasture. And that's important because the species in these two groups really have a role in fiber fermentation. So we see the decrease in these fibrolytic bacteria when horses move from pasture to a diet that has more concentrate in it. This study also looked at groups of bacteria that were more associated with degradation of things like starches. So for example, this Bacillus lactobacillus streptococcus group over here, and we see that the relative abundance of this group of bacteria was greater in horses fed concentrate versus horses that were maintained on the pasture. And this is important because these species, not only are they capable of degrading sugars and starches, but they also tend to produce a lot of lactic acid, and they can be associated then with a reduction in pH in the hindgut. So I've just talked to you about community structure and composition, diversity, species composition. But what about the function of those bacterial communities? So there's two different ways that studies can look at this, or that studies have traditionally looked at the function of the hindgut bacterial community. And the first are kind of traditional culture-based functional assays. And the second way is to use metabolites from microbial fermentation, primarily those short-chain fatty acids that I talked about before, as well as things like lactate, as markers of microbial community function. And that's much more common in more modern microbiome studies to use those metabolite markers. And so here we're going back to the study with the grass-fed versus the horses that had concentrate in their diet. And they did this here. They profiled a bunch of different volatile or fatty acids or short-chain fatty acids and lactic acid. And they found that lactic acid was greater in the horses that were fed concentrate versus the horses that were maintained on pasture. And so that tells us, kind of what we saw in that species composition and that increase in relative abundance of, say, Streptococcus did result in a functional change in the hindgut. Okay, so we've talked about the bacterial community, structure, composition, function, but what does this actually mean for the host or the horse? Because so far we measured things about the bacterial community, but we really didn't measure anything in the horse itself. And there's a lot of different conditions or different parameters you might be interested in the horse, but the things that are most relevant to our discussion tonight are going to be things like weight gain and metabolic measures. And so studies have begun to do this. So this is called a network map, and your eyes may be going every which way. And don't worry, you don't need to know what each of these little points are or what they mean. But in this study, they evaluated relationships between blood analytes like insulin and different microbial groups. And what was really interesting in this study, if you look at insulin here, when you read these network plots, usually the lines are going to tell you something. So there's a line between a group of bacteria, which in this study they designated with these 2-letter codes, and whatever, say, blood analyte in this instance you might be interested in evaluating. And these lines are color coded. So in this case, If there is a negative correlation, the lines were blue. And if there's a positive correlation, the lines are red. And if you look at insulin here, you see just a lot of blue lines coming off of insulin. And in fact, they found over 50 bacteria that were negatively correlated with circulating insulin in horses fed a variety of different diets. More recently, this has started to get even more attention. So there's been studies that have evaluated the relationships between the gastrointestinal microbiome and changes in weight, such as weight loss and weight gain. And moving from that to trying to understand, are there any relationships between that hindgut microbiome and some of our metabolic responses, specifically glucose and insulin? And the reason that this is of key interest is because if you look at the human literature and some of the laboratory animals that have been evaluated for human metabolic conditions, things like type 2 diabetes, the literature on that is pretty strong. And what they found in those studies is that the onset of metabolic dysfunction in people or in some of these laboratory animal models that they utilize is actually microbiome dependent. So that means the changes in, say, your glucose response to a meal are actually mediated by your gut bugs. We don't know if that's necessarily the case in the horse. It's really difficult to determine causation in these studies, but there are a number of studies recently that have started to look a little bit deeper at this concept. And so that brings me to some of the research that I'm going to share with you from my PhD work. So in my PhD, we were really focused on this concept of integrated warm season and cool season grass rotational grazing systems. And we looked at this from a lot of different angles on production and grazing behavior and metabolism and the microbiome. So we're going to focus in on kind of that relationship between the metabolism and the microbiome of horses grazing warm versus cool season grasses. And just a little bit of understanding about what this is, because I think it's hard to understand how we set up our experiments and why we made some of the choices that we did if you don't understand why we would want to do this to begin with. And so cool season grasses, things like Kentucky bluegrass, orchard grass, tall fescue, that are commonly grown in temperate regions of the United States, they have this characteristic peak in growth in late spring into early summer. Another secondary peak as cooler temperatures return in the fall. But they also undergo a period of low productivity during the hot, dry months of the summer. Conversely, warm season grasses are most productive during that same summer period when their cool season counterparts are semi-dormant. And so by developing a grazing system that incorporated both warm and cool season grasses, we could bridge that summer slump forage gap and potentially increase season-long production. And as a side note, in addition to these differences in seasonal growth patterns, we also see differences in nutritional profiles between warm season grasses and cool season grasses, at least characteristically. Warm season grasses are considered to be lower in non-structural carbohydrates, or NSC, than cool season grasses. What does this look like in real life? So I've just shown you a graph, but sometimes it's hard to visualize, and sometimes a picture is worth 1,000 words. So this is a picture of two sections within our crabgrass integrated rotational grazing system. So we planted crabgrass, which is a warm season annual, in the section on the left, and this is our cool season grass in the section on the right. And from this picture, which was taken on a very hot, dry period, I think it was 90 degrees on the day that this picture was taken, it's pretty obvious to see that that crabgrass is much more vigorous and productive than the mixed cool season grasses in the section on the right. So again, going back to this, when we're talking about the impacts of any difference in diet, in this case, warm season grasses versus cool season grasses, on the microbiome, there's kind of two different ways we want to look at these interdiet comparisons. Just as a reminder, these differences after adaptation and then fluctuations when we're transitioning between two different feedstuffs. And we're going to start by talking about how we evaluated and what we found when we looked at horses after they've been adapted to the warm season versus the cool season grasses. So in this study, we took 8 healthy standard bred mares, and we randomly assigned them to one of 2 grazing systems, and then collected fecal samples following 2 to 3 weeks of adaptation to a standardized hay diet in the spring, cool season grass pasture in the spring, warm season grass during that summer period, and then after return to cool season grass in the fall, and finally following a standardized hay diet at the end of the grazing season. We also conducted oral sugar tests following the cool season grass pasture in the spring, the warm season grass during that summer period, and hay in the fall. And we're going to start there with our oral sugar test. So we're going to start with that metabolic measure. And so what we found here was that there was a difference in our area under the curve and our peak plasma glucose in response to oral sugar tests in these horses. So we did see that both the area under the curve and the peak plasma glucose actually was lower in horses that were adapted to the warm season grass versus when horses were adapted to either the cool season grass or the hay diet. However, we did not see these differences in glucose recapitulated when we looked at insulin concentrations. And furthermore, just to be clear and transparent for all of you at home or maybe aren't familiar with what the reference ranges are for circulating glucose in horses and what acceptable ranges are in response to oral sugar test administration, these levels, regardless, yes, there was a significant difference between treatments, but these levels are considered normal. None of the horses in our study kind of fluctuated outside of that normal range. And then we evaluated the microbiome. So we started, just like I showed you in the background literature, we started with diversity. Okay, so, and what we did find, this is alpha diversity. So species richness and evenness actually was greater in horses when they were adapted to the warm season grasses than when they were on cool season pasture, either in the spring or the fall. And then we wanted to look at species composition. So we took our total microbial community and we grouped them by co-abundance. So bacteria that were either increasing or decreasing between the different grasses together. And we did some fancy statistics and we whittled that large number of bacterial co-abundance groups, that 333 BCG that we found, down to the 25 groups that were most influential, that were most influenced by forage type. Okay, so we found 25 groups that were most influenced by forage type. And we confirmed this through some more fancy statistics. And this random forest classification, that sounds like a big word, but the really cool thing about this is that what we did through this modeling approach is we were able to predict forage type based on the abundance profiles of these 25 bacterial groups at an accuracy of 0.9. And for reference, the best you can get in these analysis, perfect accuracy is 1.0. So this is actually very high. And what this means, if this number is high, yes, we could predict forage type based on bacterial composition. But the opposite then is true. So that means that forage type was influencing our hindgut microbial community composition. We then did some more analysis to look at taxa in these bacterial groups that were specific to each of our individual forages. We don't have time to go through every forage and what we found, but one interesting thing that we found was that in horses that were adapted to the warm season grass, or when horses were adapted to the warm season grass, we did find that Akkermansia and Clostridium butyricum were identified only in those bacterial groups that were markers of the warm season grass, that were enriched in the warm season grass. So I just talked to you about, okay, we did our diversity, so our community structure, we did our species composition analysis. Okay, function, and we use that approach of markers, metabolites as markers of microbial community function. And really what we found here, again, here we have a table, lots of numbers, lots of things. You don't need to worry about all of the numbers. That's not the important thing, at least for purposes of this presentation. What is important is that where we really found the differences were between the hay diet and the pasture diet, regardless of which pasture samples we were evaluating, their difference was between hay and pasture, whether that was for our short chain fatty acids or some related metabolites called branched chain fatty acids. It was really differences between the hay and the pasture. Although I would point out that while there was no statistical difference, numerically, the concentration of pretty much all of these fecal metabolites was intermediate between our cool season grass pasture and our hay diets. So then we sought to understand, is there a relationship between the microbiota and the nutrients in the pasture forage? And so we did kind of a similar approach, that random forest modeling that I was talking about before. And instead of saying, can we predict what forage type, we want to determine, can we predict nutrient concentration? And actually we found that we could, but we did find something kind of surprising. So we would have expected that we would get a high accuracy for fiber, because fiber is important in forage, right? And we know from other studies that fiber is a key driver of a lot of things. But the accuracies were actually highest for crude protein and our non-structural carbohydrates and our water-soluble carbohydrates. So then we did a little bit more evaluation of these relationships between the nutrients and the microbiota. So I want you to remember that we said Clostridium butyricum and Akkermansia were specific to warm season grasses. So these two groups of bacteria, this BCG 113, BCG 35, they were enriched in horses when they were adapted to the warm season diet. And these also happen to be positively correlated with crude protein and negatively correlated with NSC and WSC. And really interestingly, when we analyze relationships with our glucose metabolism in these horses from those oral sugar tests that I showed you first, this group with Clostridium butyricum in it actually was negatively correlated with peak plasma glucose. So what does that mean, negatively correlated? And that means as abundance of this group with Clostridium butyricum increased, there were actually decreases in peak plasma glucose. So what is kind of the meaning of all of this, and how do we kind of interpret these results for community structure, composition, function, and our relationships between forage nutrients, the microbiota, and metabolic responses? So first off, I think we've pretty clearly demonstrated that there are shifts in grazing horse microbiome structure and species composition within that framework of an integrated warm and cool season grass rotational grazing system. We also found that crude protein, NSC, and WSC were the nutrients that most influenced microbial community composition in response to changes in forage, not fiber. And again, I said this is curious because high fiber diets have previously been associated with greater diversity and reduction of lactic acid producers and less acidic pH. I showed you one of these studies earlier in the background literature. However, it's worth noting that most of these studies that have found these effects of higher fiber diets have primarily evaluated diets that included concentrate versus diets that did not. And here we were just evaluating differences between two forages. And so perhaps there is a minimal shift in the bacteria that are most responsive to fiber above a certain concentration threshold. So warm season grasses, cool season grasses, both of them are pretty high in fiber. So perhaps they have a high enough plane of fiber that those bacteria that are most responsive to fiber aren't really affected that much by changing between the warm season grass and the cool season grass. I should note that we didn't in the study evaluate nutrient digestibility, so that also could have played a role here. We also found in the study that differences in fermentation metabolites were primarily between the forages and the hay diets, and this could be reflective of functional redundancy in our bacterial communities. So what this means is that, yes, we may have differences in species composition, but perhaps those species are performing the same function. There are a lot of different bacteria, even though they are in different genuses, different families, they still have the capability to perform the same metabolic functions and therefore potentially produce the same fermentation byproducts. And finally, we found that Akkermansia and Clostridium deutericum were enriched in warm season grasses. And this is important because these two groups, or bacteria in these two groups, have been previously associated with healthy gut function and have been investigated for probiotic use in humans and other, livestock species, but have not received a whole lot of attention in the horse literature. However, recently, Akkermansia species have been shown to modulate inflammatory responses in horses. And we know that a lot of times in these relationships between the microbiome and metabolism, especially between microbiome and metabolic dysfunction, we see inflammation kind of as a mediator between the microbial community and the response of the host. So we talked about the differences after adaptation, but we should just briefly touch on what happens as horses transition between warm season grasses and cool season grasses. So here we have a pretty simple experimental design. We evaluated horses during transitions from cool season grass to warm season grass, or CW transitions, and warm season grass to cool season grass, or WC transitions. And we collected manual grab fecal samples on day 0 to 4 and 6 of each of those transitions. And we start off with diversity where we started before. And remember, with these PCOA plots, what we're looking for is clustering. So in this case, if we were going to see differences by day, we would see all of the smaller points clustered in one part of the plot, and maybe all of the medium-sized points clustered in a different part of the plot, and all of the really big points, whether they're a triangle or a circle, clustered in a different part of the plot. And we don't see that here. There is no clustering by day, which tells us that there really probably isn't a distinct microbial structure change across days within either the warm season to cool season or the cool season to warm season transitions. We also looked at species composition. So we did kind of that same fancy statistics to determine if we could predict day. Remember, when we were predicting forage before, we had a value of 0.9. Here we have a value of 0.25. So again, some more evidence that day was not really influential, that we didn't see big shifts by day in species composition across these transitions between warm season and cool season grasses. And this is especially when we compare to the predictive accuracy for things like the grazing system that the horses were grazing in, the specific transition, CW versus WC, or even the individual horse. All of these were well above 0.9. And so day really wasn't that influential. And this was confirmed when we did differential abundance testing on this. So we looked at the relative abundance of these species across days, and actually only 3 groups differed across days. And this was less than 1% of the total microbial community. We also evaluated pH. So we didn't do the full suite of metabolites for this part of the study, but we did use pH as our marker of microbial function. And while there were some slight variations in pH across days, there were no statistical differences regardless of transition. And again, similar to what I told you for the glucose measures, none of these values for pH are out of a normal range, and all samples, in fact, were above 6.5 for all horses regardless of transition. So from this, we can determine that the microbiota really were relatively stable across transitions between warm season, cool season grasses. And this integrated rotational grazing strategy probably can be implemented without any concerns about these fluctuations in the microbiome, potentially inducing some kind of dysfunction. I will note, however, that when we analyze the nutrient composition in our grasses, our warm season versus our cool season, we actually didn't see as much difference as we would have expected to see. I said there are characteristic differences, but there are so many factors that can influence what the actual concentration is in the grass, right? And so I tell you that, yes, you can transition between warm season grasses and cool season grasses without having to worry about the effects on the hindgut all that much. But Dr. Williams will tell you in every one of her programs that you should have your pasture tested. You should know the nutrient composition of your pasture if you really want to do a good job of managing your pastured horses. And if you're going to implement a system like this, this is very true, because if you see large nutritional differences, we would expect that potentially you would see something different reflected in the hindgut microbiome. And finally, if you take all of this together, what I showed you from the adaptation study, what I showed you from this transitional study, really what we see is more of a gradual adaptation of the microbiome to these differing forages, right? We did see differences after horses were adapted, but those weren't really apparent during that transition period. So just some final thoughts on everything from kind of our background through the research that I shared with you from my PhD. What does all this mean? Okay, what does all this mean? When you come away from here, what should you know? So there are distinct but subtle differences in microbial community composition, all right, between horses that are consuming different forages. That's pretty much established, and this has been shown in other studies, specifically differences in horses consuming, say, alfalfa hay versus grass hay. And there are functional differences between these communities. So not just differences in the community composition of the microbes, but what they're doing in the horse. We also find that grazing horse microbiome is responsive to more than just fiber. And this is important when we start to think about, the nutrients that we supply to our horses in pastures and what we consider important in evaluating. And then we've started to establish that there are some potential relationships between the horse metabolism and the hindgut microbiome. There are some associations there that should be further explored. And finally, I'm going to tell you more research is needed. We pretty much all say this for everything, but I think I can illustrate this to you really well. So if you go to this, if you go to Google Scholar on the internet, so you can go to Google Scholar and go to the website. This is a search forum, so you can search all kinds of studies on all kinds of topics through Google Scholar, excuse me. If you could type in hindgut or gut microbiome in Google Scholar as your search term, so just type that in there. When I did this in 2019 for a similar presentation, there were 169,000 results. When I did this for this presentation, 563,000 results. So this is gut microbiome, regardless of species. This is all species, any species, including humans. Gut microbiome. So there is a lot of intense interest in this area, right? And it is growing at a very fast rate. And what we are learning about the microbiome is growing at a very, very fast rate. So let's now go and search equine hindgut microbiome. I want you to think about, what do you think happens when we search equine hindgut microbiome? How many studies research results do you think, how many studies do you think come back in the results if you search equine hindgut microbiome? Yeah. 458 results in 2019, 801 in 2022. So a lot less than we have for other species. And this means that we know a lot less. We have a lot farther to go in understanding the composition and function of the hindgut microbiome and how that relates to things like metabolism. But relative to the size, this is still a growing field in terms of equine research. So we see, on a percentage basis, this is something like 75% growth in this area just in 3 years' time. So we said more research is needed, but we need to make sure when we're talking about this research that we go through all steps of this. So these studies need to not just include analysis of microbiome composition, but we also need to analyze the function of that microbiome. And we need to determine, does that microbiome respond to diet or other variables that we might be interested in? And then does the horse respond to the diet or other variables? And then kind of marry those things together to determine if there's a relationship between those microbiome and those horse responses. With that, I'm gonna wrap things up. I want to acknowledge all of our all the people who helped make my dissertation research possible, especially the Rutgers Equine Science Center and the New Jersey Agricultural Experiment Station for all of their support during my doctoral program.
Excellent talk, Dr. Weinert. Jen, it's still hard for me to call you that, but congratulations on a wonderful talk. There are a couple of things, if you don't mind, just leaving your slides up, because something did come up and I wanted you to finish first, but a couple of people caught the word Shannon on some of your graphs. Could you just talk about briefly what Shannon means without getting too technical?
Yes. So Shannon is a index that is used to measure species richness and evenness. And that's probably not going to help you because you're probably still like, I have no idea what you just said. But I have a couple slides that might help with this concept. I didn't have time to put them in the actual presentation because we do have another wonderful presentation on hemp coming up in just a minute. But I did throw these in here. So if I can get to where here. So Shannon is a measure of alpha diversity. So what does that mean? Alpha diversity. It's kind of really, and you might have heard diversity, a more diverse gut. You might have heard a supplement that is marketed as, resulting in your horse's gut being more diverse, or maybe a feeding regimen that might, make your horse's gut more diverse, or maybe even differences, say, between lean horses and obese horses. And yeah, there's differences in diversity. This is usually what they're talking about. They're usually talking about alpha diversity in measures like Shannon. And these pictures really illustrate it well. So I know you're looking at flowers, you're not looking at horses, but bear with me for just a second. So you've got this picture as field of tulips on the left, and then you've got this beautiful, colorful garden with a lot of different kinds of flowers on the right. So it's pretty easy to say that this garden on the right, this really, really colorful garden, is more diverse than this field of tulips on the left, right? We have more species, more richness of species in this multi-flower garden than we do in the field of tulips. So that's really what we're talking about with Shannon and alpha diversity in a nutshell.
And I didn't plant that question, I promise. I didn't realize that it fit in so well. Well, I did ask Dr. Weinert to go through some of the Q&A, if she would, while the second speaker might be speaking. So some of the questions can get answered there. There might be just one more that I would like you to answer if you could quickly, and one that probably is good for everybody to hear. Because as we know, our horses are selective grazers, and a lot of them prefer one, maybe two spots in the pasture over something else. So if our horses did this in the study, which we rotationally graze, so it was a little more limited, but if we allowed them to do this, would this affect their hindgut microbiome in any way? And if so, how do you think that would do so?
So I think it all comes down to the nutrients that are in the grasses. So we did evaluate representative samples from all of our pastures. And so for us, I don't think that it was that selection is probably necessarily a huge deal, because we just didn't see that much difference in nutrient composition regardless. And I could go into my whole dissertation on how we really didn't find much in differences in nutrient composition, at least not as much as we had expected. But you are right, they are selective grazers. So if you had, say, a pasture that had one grass that was really, high in non-structural carbohydrates, and it just happened to be that your horse preferred that grass more and grazed that grass more often, yeah, I would expect you would see more differences, because there's a pretty, good indication that those nutrients are pretty influential on the hindgut microbiome of horses. And that's kind of inconsistent across the literature, regardless of if it's grazing horses or horses that are fed, say, concentrate diets. So yeah, selection could impact it. I don't think it was a huge impact in our study because horses didn't have the choice in this case to graze warm season grass versus cold season grass. They had either warm season grass or cool season grass, and they were only allowed access to that one section containing that one specific kind of grass.
Excellent. Well, again, if anybody has any additional questions for Dr. Weinert-Nelson, please go ahead and ask them in the chat. I did ask her to go and just peek through the chat and see if she has any she wants to answer now, but we will have plenty of time at the end, and we'll revisit this and we'll answer some more questions later. So right now, I would like to ask our next speaker, Anna Drager, to go ahead and pull her video on and go ahead and share her screen. And I'm going to introduce her. So I will say, in part of my introduction, I'm going to explain how we came up with this topic because I knew I wanted Jen to talk about her current research. And I was really trying to think of what else is really new and really upcoming, and what kinds of nutritional stuff have we done that I get asked about a lot. And in brainstorming with Jen, we said, you know what we don't know a whole lot about ourselves, but I would love to know more about is using hemp and CBD and these types of supplements in horses. And I know I get asked a lot of these questions, and there are a lot of things I can't answer. So that is why we targeted this topic. And in my search, I happen to know Dr. Shapur, who Anna worked with for her master's project. So let me give you a little bit more about Anna and go full circle here. So Anna is a graduate of Murray State University. As a student at Murray, she obtained both her bachelor in equine science and her master's in agriculture. She was a recipient of the Outstanding Graduate Student in Agriculture Award, so congratulations there. And following her graduate assistantship, who was under the mentorship of my good friend and colleague, Dr. Shapur, she actually works a lot as an Icelandic horse trainer. And I was commenting on her very cute picture when she was off her camera. She traveled from New York to Thor Icelandics, and then down to Georgia at Creekside. She currently works for Tapter Icelandics LLC in Crestwood, Kentucky, where she trains horses, teaches lessons, creates educational resources for horse owners. So today she's going to give us the latest and greatest information all about her work in CBD and hemp in horse nutrition. So go ahead and take it away, Anna.
Thank you, Dr. Williams, for the introduction and for allowing me to be a part of this webinar series. So today I'm going to take you guys through kind of a summary of some of the literature that's out there on both cannabidiol and hemp overall, not just specific to equine, and then we'll focus in on my master's project that was specifically on equine nutrition. And so I'm sure everyone's seen a lot of products that are out there with various claims, both in human supplements that are out there, and then also canine and horses. And these are just a few of those claims that I kind of gathered together. And the thing with these is a lot of them are actually rooted in studies that have come out that show that to be the case. But sometimes with the supplement-based products, that gets taken out of the research context. So part of what we're going to do today is go through some of these and talk about what research has found and in what specific cases that's actually helping with things like reduced nausea and vomiting, calming agents, focus and memory, and things like that. But first, kind of in association with that, I want to talk a little bit through the legality of it, because there's a lot of gray area concerning hemp, and then definitely any sort of derivative that's from hemp. And so in 1906, we had the Pure Food and Drug Act, and that is basically, it's what laid the foundation for the FDA, and that protects consumers from product mislabeling and misbranding. Then in 1937, we had the Marijuana Tax Act. So that places an excise tax on the hemp product. And so that's at the point of manufacturing. And then in 1970, that's when we see the federal ban on all cannabis sativa, and then as well as marijuana. And so this is when we stopped the use of legalized marijuana, or we stopped the use of medical marijuana as well. And then in 1996, this is interesting because the state of California decides to legalize the use of medical marijuana. So that's standing against this Controlled Substances Act that was still in effect at the federal level. And then in 2014 and 2018, so our most recent acts, the Farm Bill acts, ended up legalizing hemp. So it separated hemp from from marijuana, as long as that was sitting above or below the 0.3% threshold of delta 9 THC. So that's the psychoactive component in marijuana. And then within that, so we have two primary regulators of feed approval. So currently, right now, any sort of hemp or cannabis-based product is not legal in animal feeds. As we kind of start to evaluate that as an opportunity for the hemp industry, that's going to lie with the FDA, so the Food and Drug Administration. They have the primary regulatory powers. And then we have the Association of American Feed Control Organization that doesn't actually have regulatory powers, but they are able to provide state-specific regulations and advice on those feed products. And then what's really important to note is that a lot of equine organizations following this 2018 Farm Bill Act kind of stepped out and made a stance against any sort of hemp product, so things like cannabidiol, definitely THC, on kind of a no tolerance stance. So those are prohibited in competition for organizations like the United States Equestrian Federation, AQHA, APHA, FEI. So several of them came out in a stance against having that in competitive horses. And the USDF explained this in a public statement. And their primary reasoning was twofold. So first of all, at this point in time, we hardly had any research, especially on horse-specific dosing of products like CBD. So we don't know how often we need to dose them. We don't know for what reasons we would be dosing them. Definitely whether or not that's something that's going to be harmful to the horse. So that's from a welfare standpoint. And then secondly, there were claims out there, as I'm sure you've seen, like we talked about, that it reduces pain in horses and reduces inflammation. And so in that terms, it could end up becoming a performance enhancement drug as well. So definitely reasons for these organizations to step up and maybe pump the brakes on allowing that in competitive horses for now. And now we're going to kind of jump over to hemp as a livestock feed. And so this is a relatively new study by Kleinens and colleagues, and they were one of the first ones to actually take our industrial hemp. So that's cannabis sativa L, as defined by Carl and Elias, and that's below that 0.3% THC threshold. And so they took the industrial hemp and they separated it out from whole plant, flowers, seed heads, leaves, chaff, stalk, extracted flower, and analyze the nutritional component of each one of those pieces of the plant. And the summary from this data that we ended up kind of coming to a conclusion with is that the seed head is probably the most nutritional component in terms of livestock feeds. But overall, as an equine feed, it's probably not really ideal. It's the level of cellulose that's in most of the plant makes it more suitable to kind of direct it more towards our ruminant species, so like cattle, for example. And that's especially the case because these are our end-use products, so they already have a market for these, for our hemp producers. And it would be more ideal if we could kind of broaden that market for our producers that are investing a lot of time and energy and financial resources in hemp to have that go into our byproducts. And then, so the other interesting component of this is that seed head that we were talking about being the most kind of nutrition containing part of it. Also has the omega-6, omega-3 ratio that we've seen kind of in human nutrition studies as being supportive of reducing inflammation when that ratio is low in the diet. So kind of a 2-to-1, 3-to-1 ratio. And because that's a component of the seed itself, it's interesting because we also see these same claims in CBD, right? That it could reduce inflammation. So there are two different types of products that we might see. We might have a whole plant product that has both the seed that has this ratio in it here, the omega-6, omega-3s, and then also the flower component of the plant, which is containing the majority of the CBD. And so at what point do we know that it's the omega-6, omega-3 ratio that's reducing inflammation, or is it the CBD itself and the pathways that works through that's reducing inflammation? So that would be an important direction for study for kind of incorporating hemp seed into the diet. And then going kind of back to that Kleinhen study from the previous slide, they also did evaluate those different components of the plant for the ability to detect different cannabinoids, including delta-9 THC and cannabidiol. And what's interesting is, so we know that primarily cannabinoids are being produced by the flower and the leaves of the cannabis plant. But in this study, they also found traces of that throughout the entire plant. So say, for example, producers have to destroy any crop that's above that 0.3% bio-dry weight threshold. If we were looking for an alternative option for farmers so they don't have to destroy that crop that they've developed all the resources into, and so we wanted to throw it into livestock feeds, or at least portions of that, we can't really do that because it could have detections of THC in that as well. And we just don't know enough yet about how that affects our livestock to be able to put that into a livestock feed quite yet. So now we're going to move more towards the conversation on CBD as a therapy. And so they've been able to kind of sidestep the FDA regulation because CBD is actually classified right now as a nutraceutical product. So you can see the definition here of a nutraceutical, but that's basically a herbal substance with physiological benefits, specifically concerning chronic diseases. The issue with our nutraceuticals is kind of like the supplements here. It's huge variation in the regulation that occurs from country to country. So in the United States, for example, we have the 1994 Dietary Supplement Health Education Act. And that act basically says these supplements are the equivalent of food. And so they're not the same as a drug. That means that allows them to be sold without proof of product safety or effectiveness. Then we go over to the European Union, and these same companies might have to have a product license in order to have that product on the shelves. And so to get that product license, they actually have to meet acceptable standards of quality, safety, and efficacy for those labeled uses of the product. Those products can't be used for anything that might exert a pharmacological, immune, or metabolic action in the body. So it has to be more like trying to correct a nutritional deficit, or supporting the physiological function as it would normally happen. What we do also know through current research is that all mammalian species do have an endocannabinoid system. And so basically this is known as this master regulatory system to where the body is trying to maintain homeostasis. And so that works through a negative feedback loop. That means that it's a self-regulating inhibitory system that attempts to restore equilibrium through reduced output of a substance. And so we have research here that shows that it's involved in a number of processes throughout the body, and that includes energy balance regulation, appetite, reward and satisfaction, endocrine and central nervous system function, and reproduction. And so this whole system relies on the fact that endocannabinoids, so those are naturally occurring in the body, are reacting with our receptors that are all throughout the body via enzymes that help them maintain those interactions. And then, so our main receptors are going to be CB1 and CB2, so cannabinoid 1 and cannabinoid 2. And these are both G-coupled protein receptors. CB1, so both of these are kind of located all throughout the body. You're going to have higher percentages, though, of CB1 being located in the central nervous system, and they're working through neurotransmitters. CB2 receptors are primarily going to be located in the immune system, and so they're working through cytokine release. There are a couple others that have been identified, also G protein receptors, and that includes GPR55 and 119. But the ones that you're going to hear most about in literature are going to be the CB1 and CB2 receptors. And so I just mentioned there are endocannabinoids that are naturally occurring in the body as well. So there's two that we've kind of isolated and identified. There are several more in the body as well, but these again are the ones that are going to be most commonly found in the literature right now. And that's anandamide and 2-arachidinyl glycerol. And these are both endocannabinoids that are competing for or acting in the same and similar fashion that THC might, that is a phytocannabinoid. And what's really cool is they've actually had a couple studies come out that have looked at endocannabinoid tone and that's in its relation to anxiety. And basically what they're starting to see now that they've identified these two is that as we see those endocannabinoid tones drop a little bit, usually there's an increase in anxiety type behaviors. And so these have both been in mice studies, so it's not in human medicine yet. But they're starting to look at this really primarily for PTSD patients in humans. But you can see how observing this relationship that might possibly be there, then we could come in with phytocannabinoids if we have an imbalance in that endocannabinoid tone and try to restore that equilibrium with a little help from plant-based products. So our phytocannabinoids, we've already talked a lot about them, but the psychoactive one is delta-9 THC. And then we also have cannabidiol, and there's like over 86 other identified cannabinoids. So you might hear cannabigerol, cannabrocamine. And there's a lot of different ones that are out there that you can find in the cannabis plant. And then what's also really important is that you realize the synthetic class is a whole different category. So synthetics are highly purified forms of these cannabinoids that we've already identified. So for example, there's only 2 FDA approved synthetic versions. And so these are both actually THC derivatives, nabilone and dronabinol, and they're used to help with appetite for chemo patients as well as HIV and AIDS patients. So the important differentiation here is these have gone through FDA approval and they have the research backing them for that specified use. There's also another product that's a THC CBD derivative, and that's Sativex. That's been approved in Canada and parts of Europe, not quite approved yet in the US, but they're working on that for MS patients as well as rheumatoid arthritis patients. And to better understand what ways we can use this as a therapy in horses, it's important we look and see where these receptors are specifically located at in the brain, because that can give us a little bit of information about what we might expect to happen with playing with those different cannabinoid levels. And so all the areas in green here are areas that we've identified receptors in the human brain. And cohesively, these all come together to influence behavioral processes, emotions, voluntary movements, and memory. So those claims that we saw in the beginning of the slideshow are not necessarily too far off. I really want to focus in on the limbic system because that tells us there's a whole nother pathway that starts to act on because it's involved in the endocrine response to emotional stimuli. So think in terms of epinephrine release and how that might affect reactivity, right? And our endocrine system might also influence mood by the release of serotonin. So this kind of opens the door of possibilities in terms of assisting us in behavioral reinforcement, especially as we start to look at that when you hear like advertisements for calming the horse in behavioral studies. What's also important is where the receptors aren't located. And so that tells us a little bit about product safety because we know that in humans, we haven't found them yet in the medulla. And that should keep the risk of overdose pretty low because the medulla in the brain is responsible for autonomic features such as breathing and heartbeat. So compared to other substances, you're going to have a little bit less of a direct risk, But remember, we just talked about its effect in the limbic system. So you might be able to affect heart rate through different pathways. For example, if we have an increased release of epinephrine, that can certainly increase heart rate, but you're not going to have that direct risk that you might have with other substances. This study by Chiocchetti and colleagues in 2020 was actually specifically in horse brains. And this was neat because they identified cannabinoid receptors in the sensory neurons and the satellite glial cells of the dorsal root ganglia. And so what that means, these areas house nerves that relay sensory information to the spinal cord. So that helps us start to understand how we might be able to impact the horse's sensation of pain through therapies. And this is what we're going to see a lot of like equestrian organizations would probably be concerned about, because if we're blocking that sensation of pain, that's certainly a problem in our performance horses. So as we started to, and I'm kind of moving now in the direction of the setup for our study at Murray State, and we really started to gain interest in this issue of chronic pain and the ability of CBD to possibly fill in as a therapy or a long-term treatment option. So there are some studies that came out in rats throughout the years that definitely showed a reduction of joint inflammation and markers like reduction of released pro-inflammatory cytokines. So this is definitely hopeful in terms of the applicability of using CBD as a treatment for specifically in terms of osteoarthritis. So they had induced osteoarthritis in both of these models and saw these benefits with CBD administration. Then we saw this study in dogs in 2018 by Gamble and colleagues, and they saw reduced pain reactions in canine osteoarthritis when they were dosing dogs for a little bit on CBD. And then in 2019, there was a study, it was a case study. So this is just a single horse, but they were looking at pain modulation in the horse. And this horse presented with signs of mechanical allodynia. So it's really hypersensitive to touch. And even like a light touch is eliciting a pain response that's way too high for that level of pressure. And so they ended up dosing this horse with 250 milligrams of CBD orally twice a day for an extended period of time. But what's interesting is they saw a complete stop of any sort of signs of pain in this horse within 36 hours of the first administration of that double dose. And so they kept the horse on this for a period of months. The horse continued to maintain that level of improvement. And then they decided to pull back from that treatment. And basically this test if we fix the problem or if we're just kind of covering up the problem. And unfortunately, it was the latter. So that it's telling us from this case study that you're looking at a long-term treatment to kind of manage pain. You're not actually solving the issue. But they were able to put the horse back on that 250 milligrams twice a day. And then slowly over a 2-month time frame, step the horse back to 150 milligrams just once a day. And that maintained a 90% reduction of symptoms. And so there's the second thing that we can kind of take from that is that even if we have to start with a higher dose, there maybe is the possibility this is either accumulating in the tissues or assisting pathways in a way that helps us pull the horse off of a full dose rate, even if they have to stay on it for possibly the rest of their life. And then in terms of behavioral therapies, so we talked earlier about kind of trying to create a little bit more of a market for hemp producers, and maybe behavioral therapies are an option for them in CBD if that's going to be, if it's able to work as it's being advertised, because we see in overall supplement use, horse owners definitely use that. So in this survey by Swersley and colleagues in 2017, 84% of horse owners said that they were using supplements for their equine companions. 47% of them said they thought those were helpful for behavioral issues. And so the current therapies for behavior. So trying to get horses to calm down, maybe for trailer loading, maybe for stall rest, are tryptophan and magnesium. Unfortunately, the literature that's out there doesn't really support it as a steady method of treatment for behavioral issues. So it's very variable in its success rates. So if we could pull CBD in as a behavioral therapy that was very consistent and reliable, that might be a whole nother market for producers. And so stepping into this study, we have some primary issues kind of at a glance here. And so one, we're sitting in an unregulated market, and that's a problem because we don't know about product safety. We don't know, there's not really any sort of regulations or standard to hold anyone who's manufacturing these CBD-based products. And so product safety is definitely a concern. And then furthermore, there's not any use recommendations out there. Are we dosing the horse once a day? Are we dosing them twice a day? How much is too much to dose them? Is there too much that we can dose them? And what specifically should we be using it for? And then furthermore, are there any side effects from either extended treatment or high dosing? And so for our study, we actually used, it's a full spectrum CBD product. So that means there are multiple cannabinoids present in the product. And it was from industrial hemp. So they actually grind down the whole hemp plant. And that's where you see the 20% plant protein, 30% insoluble fiber. And then we have some flavoring agents and a mold inhibitor that's present as well. And so this is a 0 THC product, and it was said to have 25 milligrams per tablespoon scoop of CBD in it. And so this is actually manufactured by Folium Biosciences. So for our first project, this really just served as a pilot project. And we were looking at what timeframes are we able to see CBD pop up in the blood, and are the current doses for the horse, so 25 milligrams, that was just for like your standard 500 kilogram horse, is that going to be enough to show up in the blood for us to set up, for example, a pharmacokinetic study to see that consistently through the time blood draw. And they also had two options for us. So EDS Farm, they had this PCR oil, and then they also had the full spectrum pellet. And we had a couple of considerations of whether we wanted to look more at the consumer and what they might want to use. So horse owners oftentimes, you know, a pelleted product would be really easy to top dress the feed with. On the other hand, the oil product definitely could be easier to get a more exact dose for something like a pharmacokinetic study. But we ended up dosing the horses at hours 1 and 2. So we only had 2 horses. So one horse got the oil and one horse got the pellet. And this is what we found. So your standard lower limit of quantification, so that's the level that's considered reliable for your detection number, is going to be 0.1 nanograms per mil. And so you see in that first hour, we were a bit below that. They have made detections down to 0.05 nanograms per mil, and you can see that here. But we went ahead and raised it up to that 0.1 just for consistency's sake. And by 2 hours, that 25 milligram dose was enough in both products to show up in the horse's blood. In both situations, we were seeing the pellet pop up higher, and because we thought from a practicality standpoint, consumers were more likely to use that, that's the product we carried with us through the rest of the project. And so in project 2, this was our pharmacokinetic study. So we had 18 quarter horse geldings in the project, and we dosed them at 50, 100, and 250 milligrams. So you'll notice we went ahead and bumped that low dose up from 25 to 50 because we really wanted to make sure we were seeing time points consistently. And then we took blood samples from catheters at a half hour, 1 hour, 2, 4, and 12 post-treatment. And this is what we found from that. So you'll notice there are not 18 time points found here. And that's because we actually only saw our 250 milligram group consistently go above the 0.1 nanogram per mil concentration that we were taking forward as our lower limit of quantification. And there was one horse from our 100 milligram group that popped up within detectable levels consistently. And so that was kind of an interesting find. And we did find palatability to be an issue, because you'll notice there's only 5 here from that 250 milligram group. So that was just because the 6 horse didn't finish their product. They only consumed about 120 milligrams of the product, so they didn't ever reach those detection levels. But this graph does, we were able to tell a little bit from this, right? So in time of maximum concentration for the time frames that we drew, 2 hours was the time of max concentration for us. And when looking through other literature and other studies that have come out, this seems pretty consistent. There is the possibility, there's a study by Davis and colleagues that they also looked at a horse, but they used an oil, but they looked at it in horses and took several time points. Their time point was moved a little bit further back, about 4 hours at time of maximum concentration. So essentially what we're seeing is that oils might take a little bit longer to reach their maximum concentration, but when they get there, it actually might be a little bit higher. So you're getting a better use out of that product than the pelleted product that we had experience with. So that might be something to look for in the future, as well as the fact that you can just more precisely dose both the oil and then other products that have developed, such as for dogs, they have these soft chews now, so you can get a really precise dose. And those actually showed when you compare that dose rate across the board, so for the same body weight dosage, that actually goes above even the oil maximum concentration. So definitely a lot of areas to grow on that front, but this was a starting point for us. And some other cannabinoid pharmacokinetics that have come out a little bit past when we did our study also were in agreement. So here we can see these were all done in horses specifically. And we see this time of max concentration of CBD in the blood be about similar to ours, so about 2 hours, both in the low dose and the high dose here. And something that was interesting that I didn't talk about on the last graph, but we were detecting THC concentrations in our horses' blood samples, which is intriguing because we were supposed to have a 0 THC product. So the same thing happened in this study as well. So consumers should be aware that it might be a 0 THC product, but as we kind of move forward on the competitive scale with these organizations saying competition horses need to have, they have a 0 tolerance for things like THC in the blood. As they develop those blood testing methods, we need to make sure that they're aware that THC is showing up in those samples. And then in this study, so their time of max concentration was a little bit longer than ours, about 4 hours, but still in the same ballpark. And they were seeing with the higher dose, they are getting a higher plasma CBD concentration. So those are moving together as we increase doses. And then Ryan and colleagues in 2021 actually were able to put numbers to a half-life time. And so that was about 9.8 to 10.7 hours. So that's going to be important for people to know in terms of blood sampling. If this does start to be used as a therapy for horses, you need to know that for clearance times and things like that, as well as how often we need to be dosing. So if it's only a 9.8 hour terminal half-life, we might need to be dosing more than 1 time per day. The other interesting thing that Ryan and colleagues found was that they did an icosanoid analysis, and that suggests that CBD has an influence on cyclooxygenase 1 and 2. And so these are how our NSAIDs are working for the most part. And that's interesting because it could be both a good thing and a bad thing. So if it could be bad in that if it's really impacting COX-1, so this has a lot of good things that it brings. So it helps protect the stomach lining, for example, from the acid. So that's why sometimes you'll have ulcers in horses that are dosed consistently with NSAIDs for a long time. So if it's really acting on COX-1, not such a good thing. We're going to have some of the same negative side effects potentially that you might see in use of bute. However, if it's really targeting just COX-2, now we might have a proactive pathway that we have seen work in other substances, and might be a long-term option for management of inflammation in horses. And so the other thing that we ran during this study was a serum chemistry, and we were specifically looking at these enzymes here, so blood urea nitrogen, creatinine, albumin, and ALP. And these came from a couple other studies that had run it in other animal species, but as they had tested these animals over a period of time or with certain doses, they noticed significant changes. So we saw the same thing, but the thing to note in ours that was the same as those studies is that all of these are still within safety reference ranges. So typically you would see if blood urea nitrogen or creatinine, were to continue to rise, that might be indicative of kidney damage. But they, within our 250 milligram dose, they did consistently go up significantly, but they were within that reference range for treatment. So not an immediate cause for concern, but if we're going to dose really high or if we're going to dose really long, because that's what we saw based on time here. So same thing, we have these raising up a little bit to be significant. And so we need to keep an eye on those for future studies as well. And so for the final project, we ended up dosing a total of 24. We ended up running just stats on 17 of those. So we isolated to just quarter horse geldings. And we dosed them for 6 weeks, and we're evaluating for changes in reactivity and movement. And I'm going to skip to our behavior evaluation because basically from the movement data, there wasn't really anything we could extrapolate from the information that we got. But there were some things that we could pull from the behavior data that were pretty interesting. And so for the behavior evaluation, we attached heart rate monitors inside a barn and then took a 1-minute resting heart rate reading, walked them down the aisle, came to the startle site. So this is called a novel object test, and you expose the horse to a startle stimulus to elicit the startle response. In this case, it was opening an umbrella around the barn corner. We read a heart rate reading here and then took the horse back to the barn, had them stand for 1 minute, and got a resting heart rate after that 1 minute. We also had evaluators, both one live evaluator and 2 video evaluators that looked at it at a later time. And they score the horse's reactivity levels from 1 to 5. This is from a table that was developed by Holland and colleagues in 1996. And from that data, so the heart rate came back, there were no statistically significant changes in heart rate over time. So not completely unexpected because we know that doesn't have a direct effect on heart rate. But we did see here that our treatment group had significantly lower scores after administration of treatment than our control group. So here we had a 3.3 on average, and then that dropped to a 2.7 or 2.17 for our treatment group. So this is interesting because it does suggest that we should continue to investigate this and its ability to lower reactivity in horses. And then another side note I just want to talk about really quick is throughout the project, we didn't really expect to find this. We were just sampling the product for dosing purposes as the project went along. And you can see we had 3 different labs that we were using. All 3 were accredited institutions. And I'll highlight here, and these are the dates, so February 2019 all the way to March 2020. And batch 1, so we had 2 different batches 1 and 3, started at 9.9 milligrams of CBD per gram product. And by the end of the project, we were looking at 3.2 milligrams CBD per gram product. So that's not surprising. It's a bioactive molecule. It's going to degrade over time. But what is surprising is another accredited lab had the reading at the same time, and they came out to 10.4 milligrams CBD per gram product. So that's definitely, a little bit alarming because we need to know for appropriate dosing what that product is actually going to be containing when we give it to our horse. And then for batch 3, we look at this and it does consistently drop as we test from these different labs. But then within the same lab here, we settle and see that it starts to slowly creep up. So there's not really a great explanation for this. My only one consideration I have is that we have either testing methods are just not consistent enough between labs to be able to compare between labs. And then secondly, remember we talk about there's neutral forms of these analytes, so CBD, CBDA, THC, THCA. And so sometimes those acid precursors will convert over to the neutral forms. And so perhaps with aging and time, that's kind of what we're seeing here, but it wouldn't make sense that one lab is getting a lower reading and one lab is getting a higher reading. So that's definitely something to consider as we move forward and looking at CBD as a therapy. And then finally, there is an issue of economic feasibility for consumers. So at that 100 milligram dose for a 544 kilogram horse, so that's about 1,200 pounds. That was the dose we were using for our long-term study. That's about 0.18 milligrams per kilogram a day. When we figured that at the price of the product that we were using, that comes out to about $4 a day that you're spending on that supplement. So not too bad until you start looking at clinically effective doses. So in Philpott and colleagues, they were looking at that. This is one of those that was looking at osteoarthritis in mice. So their dose is a little bit higher here, you can see, and obviously that cost per day goes up a little bit. Still not terrible, maybe a little concerning if you're going to keep the horse on that for a long time. But then we go to the canine study. And if we were to dose them at their highest dose for this canine study, you're going to be spending $181 a day. So now we're getting a little bit alarming, right? And if we go into one of the most successful examples of an osteoarthritis case where they were able to reduce joint inflammation and cytokine response, we're up to $1,000 a day. Now here we're starting to question if we should just jump into pharmaceuticals, right? And just use an FDA approved product. That's especially concerning because we see what consumers are currently paying for products like this. So Smart Calm, so a calming supplement, we're only spending about $1.39 per day. So that's even still lower compared to that lowest price point, about $4 a day that we were talking about from our project. Then we have Smart Flex Senior, so kind of going more in towards the joint focus. That's sitting at about $1.66 per day. And then something like Farrier's Formula, this is a really common one that I'm sure a lot of people have heard of. That's only 89 cents per day. So definitely this is something to consider as we move forward in analyzing this as a supplement. We've got to find a way to make it a little bit more affordable. So implications from our project and kind of reviewing the literature is that we do see CBD uptake occurring in the horse, both through pelleted products and oil-based products. Pharmacokinetics are comparable, but detection is inconsistent. So both within species and, within the same species, we see those the time of maximum concentration being about the same, but we really need to work on developing more consistent detection methods. And then we see that reactivity was reduced in treatment horses as compared to control horses in that behavior component of the study, which that really further indicates the need to continue to research that route. And ultimately, it could become an issue of feasibility and consumer practicality and making this supplement available to consumers. So with that, I'm going to end. I'll type through my sources here. But thank you all very much for your time.
Thank you very much, Anna. What fascinating stuff. I love talking about supplements, and especially to get into the research like that. That was excellent. I will say I am gonna take questions for both of you. We've got a lot of questions that have come in, and I know Jen has saved a couple live. But right before, I know we do have a few people that probably need to log off soon. I just put in the chat, if everybody doesn't mind, before you completely finish for the night. I've been getting a few chat messages saying that they really want us to keep the webinars going, which I would love to, but I would also love for everybody to tell us what you would want to return to hear. And in order to do that, I need you to fill out the survey. So tell us what you thought of today, how you like the talks, and then if you are interested in coming back for more webinars, what do you want to hear? It's just a really quick survey, but please go ahead before you end your night and just help us by filling that out. Anna, I'll start with you, if you don't mind, with a few of your questions. And I know you briefly mentioned that there are some, or we need to investigate some potentially long-term effects of CBD. Are there any that you know of, or any in maybe other species that have been?
Yeah, so there aren't actually any that have shown up from these plant-based products, right? Now, definitely when we start getting into pharmaceutical types, they're more purified substances. But even those don't have these really negative side effects. So we haven't actually seen evidence of like kidney damage or something like that, for example. Most of the time, it's just a little bit of a nauseous reaction or something like that.
But it sounds like primarily, you got to get them to eat it first, right?
Yeah. Unfortunately, the battle of supplement studies.
Of course. Then this one's a good one too. Can horses build up a tolerance to CBD so that eventually they might need more than the dose they're getting?
So based on the information that we have so far, my inclination is no, but that is definitely an area of research that we need to look more into. And I say that because if you go back to that study by Ellis and Contino, they were actually able to reduce that dose load. So that makes me think that we're not going to head into a situation where you're having to dose more and more, because they were able to step that horse back and still have that continued benefit.
Cool, great. So what about any possible THC research, since we've been going down the CBD route? Do you see that anywhere in the future?
That could be a difficult one, because I feel like it has such a negative context right now. I actually haven't come across any that are like a strict THC test in horses. I imagine it could be if ideas kind of start changing and mentalities start changing towards it a little bit. But I think that one scares, it's a little bit more concerning, because we don't necessarily know how a 1,200-pound animal is going to react to having loads of THC, right? It could actually be a little bit more of a dangerous scenario. I don't know.
That's a good point. That would scare me, actually. That would scare me quite a lot. So we'll take one more. Well, actually, there's two more that are probably pretty quick, and then I've got a couple for Jen. So what about looking at either the hemp seed or the hemp oil? And I know you talked about it just briefly. You also talked about pain management and inflammation. Did you notice, what about any changes with respiratory variables or respiratory improvement? So seed or oil, and then respiratory improvement.
Yeah, so what specifically about the seed or oil? Or
this is when feeding hemp, should we look for either the hemp seed or the hemp oil? So which one would be your preference?
So I guess it depends on if you're going more for a therapeutic option, then you're going to be looking more for like the CBD based oil. If you're looking for the nutritional components, that's where we start to see hemp seed being involved. Does that help? Does that answer the question?
I think it does. Well, can let them chime in if it didn't, but I think that's it. And then did you notice any change with any respiratory improvement?
No, we did not. We didn't specifically measure anything like that, but nothing that was, very obviously changed occurred for us. So that would be an interesting topic, though.
Yeah, and I know another thing, and actually there's some of the at Rutgers that are looking actually at hemp as a bedding source. They're not doing it with horses, but they're doing it with some other animals. Do you have any experience with any of that research? And could there be any negative consequences with using hemp for that?
So Murray State was actually talking about starting a hemp bedding research project as well. And the only concern that I remember being brought up at that point, I don't know if they ended up doing it, but that animals might end up eating the bedding. And then again, that becomes an issue if you're using those horses for anything, competition wise, and they start testing, that's probably going to show up in your blood test. So that would be my main concern. Other than that, I don't know of any reported issues for bedding use.
Yeah, good. You do have a few more, but let me switch over to Dr. Weinert for a little bit. And if we do have a little bit more time, I can come back to them. But I know Dr. Weinert was busy answering some of these in the last little time that she's been on. So some of them have been already answered. But I will start with, does a more diverse gut health, better hindgut microbiome help a horse that has a tendency for gas colic incidents? And if so, does it minimize the risk? And in what way would you best achieve this? Are there any supplements or warm season grasses?
So this is a really kind of ties into what we were talking about at the end of the presentation and that question that we had about diversity. And this is kind of one of my pet topics that I always like to drive home in any talk or discussion that I get, because a lot of times, you know, we traditionally think, or kind of the conventional line of thought is that a more diverse microbiome is better, right? A more diverse microbial community. And there's a couple of reasons for that. Perhaps having a more diverse species composition means that your microbiome maybe is more functionally diverse. So maybe they can better access different nutrients in the diet, and they can take advantage of the full range of nutrients provided by the diet. That could be a selective advantage in certain circumstances. But the big one that a lot of people point to is there is this thought that a more diverse microbiome is more resilient and is more better able to adapt to changes in diet or, you know, other factors, say environmental factors. And, you know, there's a reason to think this, and it's a logical thought. However, I would caution everyone that more diverse does not always mean better, healthy, etc. Okay. And a classic example of this is if we think about lean horses versus obese horses. So the bulk of the literature that have evaluated the hindgut microbiome of lean horses versus obese horses, with some exceptions, have mostly found that the microbiome of obese horses is more diverse than the hindgut microbiome of obese horses. Now, I don't think that we could reasonably say that that is a marker of health, right? We wouldn't say that the microbiome of obese horses is likely to be healthier or better than the microbiome of lean horses. In fact, most likely what's happening there, what could potentially be happening there, is that microbiome is more functionally diverse and is more efficient at metabolizing the nutrients that it receives, and therefore helps to kind of exacerbate and potentiate those problems with weight in obese horses. And so in that case, a more diverse gut is actually not a good thing. In terms of colic and, you know, the relationship of microbiome and colic, there's a lot of recent papers that have started to unravel those relationships, but I think they have a long way to go. And I would caution about just thinking about diversity. Diversity isn't necessarily the key. It's the specific bacteria and not a genus of bacteria, not a family of bacteria, definitely not a phylum of bacteria, but specific strains of bacteria that could be used. Now, I know Dr. Williams answered another question in the chat, you know, about fecal microbial transfers. So there is, you know, ways that you could possibly evaluate changing the microbiome in horses without having to identify the specific bacteria that are responsible for, say, a protective effect against something like gas colic. But really just promoting diversity probably isn't going to cut it.
Good answer. So I like this one, and I'm not sure if you're gonna go there and talk about the study that didn't happen or not at Rutgers, but we use standardbreds. That's what we use at Rutgers. which, I guess compared to some horses aren't as metabolically prone, even though they can have metabolic syndrome. But what do we think would have happened if we would have used some breed that is metabolically prone for insulin resistance, laminitis, etc.?
This is a great question. So our horses, not only were they standardbreds, but they were healthy. They're healthy adult horses. They had, body condition scores within that 5 to 7 range, which is kind of considered, normal body weight, right? So they weren't even excessively fat, standardbred horses. And so we wouldn't necessarily expect to see as much difference. And definitely, I don't know if whoever asked this, and for some of you who are still on here, if you were able to come to Dr. Adams' talk last week, and she was talking about insulin dysregulation in horses and how there are differences in responses, even to the same feed stuff, between horses that are, metabolically dysregulated versus horses that are metabolically healthy. Right. So if we had horses that already had existing metabolic dysfunction, or perhaps were excessively heavy and were trending towards that kind of dysbiotic state, right, I would totally expect us to see differences, not just in their metabolic responses to different, forages with different levels of, say, NSC, but I would expect that to be reflected in their microbiome as well. There are a few studies who have, I just mentioned, some of the studies that have looked at differences in obese versus lean, but there are studies that have evaluated or are beginning to evaluate the microbiome in horses with metabolic syndrome. And there are distinct differences between horses with metabolic syndrome and horses that are healthy. And therefore, if their microbial communities are different, their responses to different diets are almost bound to be different.
Yeah, so we were going to investigate this a little more, not with the unhealthy horses, but we were at least going to use some obese or standardbreds that like to get obese or fat on grass. And COVID kind of curtailed that for Jen, unfortunately. But there's definitely a lot of area. We'll let Jen get done with her postdoc and into a nice position at another university, and then she can answer a lot of these questions for us, hopefully in the next 10 years, right?
Great. There you go. There you go.
So I'm going to switch and ask a few more to Anna, and then we might come back. I know, again, we are out of time, but I'm going to keep going because we got some great questions. If you do need to leave, please feel free, but we still have over 100 people on, so I'm sure everybody still wants to hear what you have to say. I'm going to pop in the link to the survey one more time, that if you are going to leave, please take our survey. We would really appreciate it. But where was the question? Sorry, I've been looking back and forth at all of these and I might have just lost it. Oh, is there any concern for the presence of THC in any of the hemp powder or meal? And you might have covered this a little bit, but can you clarify?
Yeah, and so this goes back to product regulation. So if it's something that's actually, if these protein meals are from the hemp seed, not so much, because the concentrations of any cannabinoid at that point are so low, and these are going to be coming from industrial hemp, so they have to be below 0.3% anyways. So I wouldn't be so worried about that. The main thing is just to make sure you're using a reputable organization when you're buying those products, I think, is the main piece.
Good. So what about any behavioral changes between horses? So I'm assuming they were housed together or housed in groups. So any of the cannabinoid horses in that treatment group, did they have any changes in their behavior together? I know you probably didn't look at it, but anything?
Yeah, unfortunately, that's a great idea. And I hope that that's an extension of the study that we could definitely do in the future. Just the way that we were managing the horses for this study, because it was over a 6-week time frame. That wasn't something we were able to look at, but definitely a good idea for the future.
Excellent. On the supplement topic, and I know I get these questions every time I seem to speak, but Jen, can you approach the subject of probiotics and how effective are they, and is there any value to feeding these?
So What is a probiotic? I think first we got to take a step back and understand what a probiotic is. So the idea behind a probiotic is that you're going to feed live microorganisms, okay, to a horse in order to alter the microbial community to get a desired response, maybe promote certain communities or promote certain functions, for example, production of certain short chain fatty acids like butyrate that are known to be protective. That is the reason or the rationale behind probiotics. There's also conversely this idea of prebiotics. So prebiotics are non-living, okay, non-living substances that you can supplement your horse that are known to impact the microbiome and again potentially promote certain bacteria that are known to be beneficial in certain ways. And is there a benefit? I think that the literature is pretty advanced, or at least more advanced in the field of prebiotics. And this is in all animal species, including the horses, this is even in humans. Prebiotics generally tend to work. And the advantage of prebiotics is that in most cases, if a prebiotic works and if a prebiotic alters the microbial community composition and function, even after you stop feeding that prebiotic, you will see some residual effect. So it's not like you stop feeding and everything that you maybe gain from feeding that probiotic just goes away. With probiotics, that is not the case. So probiotics are a tough issue. There's a lot of things that can influence whether a probiotic is effective. So functions in bacteria and whether or not they are effective or not at producing a certain response are really strain specific. So what that means is you've got to have the exact bacteria that the horse needs in order to get that response. And so sometimes, maybe you have a bacteria that is in the same genus or maybe even the same species, but it's not the right strain. And so you might not get that response that you wanted. Also, you got to have a product that's going to make it all the way through that foregut, through the acids in the stomach, etc., and into the hindgut in a live form with enough what's called colony forming units or CFU that it actually can produce some kind of effect. And there are some that have been shown to produce some effects. A lot of cases, it's not as much even the bacteria that have been shown, but there's a lot of research into use of yeast as a probiotic. And so for that, though, usually speaking, when you discontinue feeding a probiotic, whatever benefit you have is gone. So basically they're present in the hindgut. There may be some impact, but they don't colonize. They don't remain there after you've finished feeding. So there's a lot of work that needs to be done in order to get better, more consistent, more effective probiotics. I think there's just a handful of species that really have that have really been evaluated in the horse. So there's a lot of work. Make sure if you're going to feed one that it's backed up by research.
Yeah, definitely agree with that. I think, and that's why you both were here today, because I knew there was research on what you were going to talk about. So really quickly, we're going to do one more. Anna, confirm what your best guess on a CBD dose would be for maximal efficacy if someone wanted to go right now and feed it.
Oh dear. So I think from what we were seeing in the research, when we're talking about affecting osteoarthritis cases, it's going to have to be a whole lot higher than what we were dosing, or you're going to have to dose for a really long time. But in the behavior, obviously we were seeing results at 100 milligrams. So that was, I think, 0.18 milligram per kilogram body weight per day. So that was a relatively low dose, but I think you're really going to have to bump that up to probably at least, 250 milligrams to 500 milligrams even, broken up into twice per day, like that mechanical allodynia case, to see a clinical benefit in horses. So.
Awesome. Well, I'm sorry if I didn't get to your question. You can always reach out to the Equine Science Center. We have an Ask the Expert page, or you can send me an email and I can forward it to our speakers. I'm sure I didn't ask, but I'm sure you wouldn't mind answering some of the attendees' questions that you might not have gotten to today. But I want to thank both of our speakers for giving up their evening. I want to thank all the attendees for joining us. I really, really want to thank Kyle Hartman. He's the one behind the black box that says Equine Science Center. He's my behind the scenes man with everything from all three of these webinars, from any registration, technical, et cetera, et cetera. He's really been the one behind the scenes on all this. So I want to thank everybody one more time, and please take our survey, and hopefully we'll see you next time. But thank you all.